My name is Neil Iyengar. I'm a medical oncologist subspecialized in breast oncology. I'm the co-director of breast medical oncology and director of cancer survivorship services at the Winship Cancer Institute of Emory University in Atlanta, Georgia. I'm very excited to be joined today by Dr. Erika Hamilton, and I'll let you introduce yourself.
Yeah, absolutely. I'm Dr. Erika Hamilton. I'm the director of the Breast Cancer Research Program and chair of the Breast Executive Committee for Sarah Cannon Research Institute based in Nashville, Tennessee. Happy to join you today.
Wonderful. Well, this has been a very active ESMO in terms of both triple-negative breast cancer, but also the HER2-positive space. And we'll focus a bit on triple-negative breast cancer here. Before we get into some of the new trial data that came out at ESMO, I thought by asking you just to set the stage and tell us right now what your approach is in the first-line setting for triple-negative breast cancer treatment.
Yeah, so, triple-negative breast cancer is a little bit different in the first line, whether you have PD-L1 positivity in the tumor or not. So we know that for the patients that have PD-L1 positivity, somewhere between about 30 to 40% of patients typically, they have benefit to pembrolizumab. And so we give chemotherapy in combination with pembrolizumab, and we have a couple choices of chemo there. And then for patients that don't have PD-L1 positivity, the standard of care is still just chemotherapy. Most of the time, I think we tend to use single-agent chemotherapy, but we do have the option for carboplatin gemcitabine. We typically use that for patients that we're a little bit more worried about.
Yes, absolutely. And two questions to follow up. One is that in your experience, what generally has been roughly the median PFS with current standard of care chemotherapy options in that first-line setting?
Yeah, it's highly variable. We have our triple-negative patients that just do really poorly and, barely make it to that year mark of survival. And then I have several patients in my clinic that have done much better than that. But, the problem in the first-line setting is even there, progression-free survival is short, on the order of about, half a year, et cetera.
Right, yes. And what about your rapid progressors? I know certainly we see patients who relapse after their adjuvant or neoadjuvant therapy. What's your treatment approach for those patients that have relapsed within a year of their adjuvant or neoadjuvant therapy?
Yeah, that's where I often think about carboplatin and gemcitabine for those quick relapsers, because kind of universally they've gotten to taxane in that neoadjuvant space. And so A, I don't really super feel comfortable with single agent, and then B, I'm certainly not going to feel comfortable reusing something that they've just had and kind of, relapse really quickly.
Absolutely, a huge area of need for that group. So let's talk TROP2-Breast02, one of the headliners at ESMO this year. Let me ask you to sort of walk us through the trial design there and which patients were eligible for TROP2-Breast02.
Yeah, absolutely. So, we have approval of TROP2 antibody drug conjugate in the later line triple negative space. And really what this was looking at was to bring this up into the first line space, and particularly for that patient population that isn't eligible for pembrolizumab. So it tends to be the majority of our patients with triple negative breast cancer. And it was datopotamab deruxtecan, TROP2 targeting antibody drug conjugate, given once every 3 weeks, versus treatment of physician's choice chemotherapy. So pretty straightforward design, honestly similar to other trials we've seen.
Yes, yes. And speaking of other trials we've seen, we also saw the data for ASCENT-03 reported here at ESMO. Walk us through that trial design and maybe tell us a little bit about the similarities and differences with TB02.
Yeah, so, the ASCENT trial 03, we have data for Sacituzumab, Govitecan in the later line setting. We have OS there, and that actually becomes pertinent a little bit when we talk about these trials. So at ASCO this past year, we saw the combination with pembrolizumab. Positive trial, looked really great. And so this year at ESMO, we saw the PD-L1 negative subset. So very similar design. TROP2 antibody drug conjugate versus treatment of physician's choice with chemotherapy. Differences, Sacituzumab Govitecan, and we'll get into this, a little bit difference in terms of side effects, but also difference in terms of administration. It's a day 1, day 8, every 21 days, and different side effect profile between the two as well. Some other kind of subtle things in the design of the trial is TROP2-Breast02 actually included a little bit of a higher risk population. and higher risk in the terms that they could have progressed quickly from that neoadjuvant adjuvant chemotherapy. And that was excluded on the ASCENT trial. So a little bit more bad actors. It ended up about 15% of those patients on the trial were those so-called bad actors that relapsed quite quickly.
Yeah, thanks for pointing out some of those differences. I think I agree that's the population or eligibility criteria is one key difference to keep in mind. We've also heard and discussed a lot about the role of crossover in trial design, and this has been coming up in terms of a difference between the two trials. So certainly ASCENT-03 allowed for crossover because of the availability of Sacituzumab Govitecan, and as you mentioned, the OS benefit. So I want to come back to your point there. Whereas TROPION-Breast02 by default could not allow crossover, rather, because we do not have datopotamab deruxtecan currently available or approved yet in the triple-negative setting. But TROPION-Breast02 did allow for subsequent ADC. So perhaps tell us a little bit about what crossover means when we're interpreting the data and whether or not we can accurately interpret a longer-term endpoint like overall survival when you allow for crossover.
Yeah, I mean, I think honestly this is something that we always have a little bit of friction with, right? Because we want to do what's best for patients, but ultimately what's best for patients is to get new drugs approved so then thousands and thousands of patients get access to those drugs. So absolutely correct with datopotamab deruxtecan. We didn't already have overall survival for that trial set. So crossover was not allowed, meaning that people that were on chemo did not get provided dato immediately afterwards. And so that really enabled us to look at overall survival in the trial. No surprise, that was positive, which is fantastic news for patients. The reason the Sacituzumab Govitecan ASCENT-03 trial made a different choice was because they already had overall survival in the second-line setting. And so they weren't going to be held to the same bar with regulatory authorities. And so honestly, what they decided was 100% the right choice. It was the right choice for patients. They should allow crossover. And in fact, their drug is available, in that second-line space. They already have overall survival. And, previously we've seen about 80% of the patients did do that crossover. So very high. I mean, we're not talking about, 20% of patients went on to see ADC. I mean, universally, patients receive that. So I do not anticipate that we'll see overall survival with the ASCENT trial. We shouldn't expect it, and they went into that knowing it. So I think it's very important when we compare.
Yeah, totally agree. I think from a patient-centered care perspective, it's really great that ASCENT-03 allowed for crossover. But I like how you put it. We're conflicted with the scientific side, where we, of course, want to really try to minimize confounding variables. Ultimately, though, I bring this up because I think it's important. We all talk about the pitfalls of cross-trial comparison, but in a setting where you're looking at trials where one allowed crossover, one couldn't, I think we have to be very careful now about cross-trial comparison. Okay, well, thank you for walking us through the design of the trials. Now let's talk a little bit about the patient populations. You had mentioned that TROPION-Breast02 allowed for some of these early relapsers. I think that was about 15% of the population. Were there any other differences between the two patient populations that struck you? These were global trials, I believe.
Yeah, I mean, not a massive difference. I think we do see most patients with the TROPION-Breast02 enrolled outside the United States. Certainly there was Europe, there was Canada, et cetera, as well. I think that's just the challenge in 2025. We live in a global world. They certainly tried to enroll patients there. We got, quite a few, but it is a small percentage.
Yeah. All right, let's talk about the primary endpoint. Can you walk us through the PFS results for, I guess, both trials? Because, again, keeping in mind that the populations are slightly different, as you pointed out, I think it's interesting that we saw fairly similar performance of the control arm with some small differences. Let's start with datopotamab deruxtecan. What should we keep in mind in terms of the AEs that were reported in TROPION-Breast02?
Yeah, absolutely. So, I think the side effect that most people talk about when we're talking about datopotamab deruxtecan is stomatitis, so mucosal inflammation. And what makes it tough with some of these drugs is that A prophylactic steroid mouthwash was recommended as part of these protocols, but there are certain places that, don't have access to it. In fact, sometimes pharmacy to pharmacy can be a little bit variable even within a city, but there certainly are areas that prophylactic mouthwash is hard to get. So we don't have great data on exactly how many people actually received it in a prophylactic fashion. And so at least for me, practicing in the United States, where I'm going to routinely use that, it makes it a little bit tough to fully interpret the stomatitis rates. But I think, stomatitis is one we've really got to watch out for 2 reasons. One, as opposed to neutropenia, patients feel that it bothers them, and so it's a quality of life issue. And 2, in my experience, if patients really get stomatitis, it can be tough to get rid of. And so I think that prophylactic steroid mouthwash is really important. And we see this with a few other drugs as well.
Absolutely. I couldn't agree more. I think in my experience, the key has really been, and you alluded to this, is to actually use a prophylactic mouthwash prophylactically.
Right.
And starting it before those mouth sores develop.
And not stopping it at 2 weeks because you didn't get mouth sores and you don't think you need it.
Good point, yes. Yes. Now, it is an ask of our patients, of course, and let's talk about ocular toxicity. This was early grade, but can you walk us through the prophylaxis for ocular toxicity and the kinds of things that were reported with datopotamab deruxtecan?
Yeah, with the ocular toxicity, mainly it was dry eyes. And so I think it's really important when we talk about ADCs to not kind of lump everything in a class. We do have ADCs that have really problematic eye toxicity, corneal toxicity, et cetera. This really was predominantly dry eye. We've kind of done a variety of things. I find with my experience with datopotamab deruxtecan, it doesn't tend to be that big of a problem, and really kind of people are maybe needing rewetting drops, et cetera, lubricating drops, but less worrisome than some of the other ADCs.
I think that's such an important point that you raise, because we have seen other ADCs, some of which are approved for the treatment of other cancers, that, as you said, have more invasive ocular toxicity, but also in my experience, this has really been surface events that are manageable with eye drops and so forth. And I think it's helpful to have an ophthalmologist or optometrist that you could potentially reach out to, but maybe not necessary, although we know that at least annual visits or checkups can be helpful in general. Any other adverse effects that stuck out to you or that we should think about with datopotamab deruxtecan?
I mean, I don't think anything that really stood out, but again, it's an antibody drug conjugate. It does have a cytotoxic payload. You are going to want to use some, antiemetics. We certainly can see nausea, that type of thing, but no big surprises.
Yeah, I agree. I mean, I think in the triple negative setting especially, we're a little more tolerant, unfortunately, to adverse effects because of the limited treatment options. But when a trial like TROP2-Breast02 comes along, it's really encouraging to see a manageable treatment being efficacious like this. So let's look at ASCENT-03 and Sacituzumab Govitecan. I think we as a community are more familiar with the adverse effect profile because we've been using Sacituzumab. I didn't see any new signals in terms of the toxicity profile there. What was your interpretation?
Yeah, I completely agree. Sacituzumab, Govitecan, on the other hand, our 2 most prominent side effects are diarrhea and then neutropenia. So diarrhea, neutropenia, and datopotamab, obviously with the mucositis in the eye. So I think it's important to remember they're both TROP2-targeted ADCs. They both have the same payload, very different profile. So with Sacituzumab Govitecan, the diarrhea I actually find to be very polarizing in patients. It's not a neratinib or a lapatinib diarrhea or even a abemaciclib diarrhea where you can kind of guarantee that everybody's going to have some diarrhea. It's just a question of how bad it is. Sacituzumab Govitecan either have patients that don't have any or they have it and struggle a little bit. It's kind of this either or, which I find a little bit interesting. I'm curious if you had the same experience. And then the neutropenia, really patients don't feel that. I luckily haven't had any patients that have really gotten in trouble with infections and febrile neutropenia, and I've not had any deaths, although I feel like I have to knock on wood with that. But the problem can be that it's day 1, day 8. And so it can present some problems in terms of use of growth factor and things like that. You can't use growth factor between the day 1, day 8, and so we tend to use it after day 8 long-acting to try to drive that white blood cell count up for the next cycle. But I think you bring up a good point. We've had it around for longer, and so people are kind of starting to get comfortable with it.
Yeah, and I want to retouch on your point with regard to some of the initial toxicities with sacituzumab. I think, again, we're familiar with it, but I do warn my patients that usually the first couple of cycles can be a little bit rocky in terms of schedule because it can get disrupted by the neutropenia, as you pointed out. So certainly prophylactic growth factor is recommended in that setting. We don't see that need with use of datopotamab deruxtecan in terms of prophylactic growth factor, but of course we touched on the prophylactic mouthwash and the prophylactic eye drops as well. Okay, so you've taken us through the data nicely. Let's talk practically how we might implement these findings in clinic. So having reviewed these results, how do you now view the landscape? Let's assume approval of these agents in the first-line setting. How do you now envision the approach to first-line treatment of metastatic TNBC?
Yeah, I mean, so first off, it's great to have choices, right? Patients love having choices. We love having choices. They're two great drugs. I think we have a choice in the PD-L1 negative subgroup now with, not yet, but we will have a choice with datopotamab or sacituzumab govitecan. In the PD-L1 positive subset, we still have that data from ASCO with sacituzumab govitecan in combination with Pembro. So still, when I have somebody with metastatic first-line triple-negative breast cancer, I need to get that PD-L1 test, right?
Mm-hmm. Yeah.
And then that's my first fork in the road. And then I think, honestly, we have the choice between both. I can see myself using both, actually, in some different scenarios. You have a different schedule, you have a different side effect profile. We can bring up some other differences within some of the endpoints of the trial. I don't expect to see OS with sacituzumab govitecan. That actually doesn't influence me in the first-line setting at all, to be quite honest, because of the data from the second-line setting. I think it's a great drug. There's clearly a benefit. The interesting thing we did see was response rate difference. Datopotamab really having a clear, higher response rate in the control arm, and we didn't see that with sacituzumab govitecan. And so I think that left some people kind of asking questions of, well, everything kind of looks similar, but why was that so different? I think the inclusion of that population that progressed quickly on chemo than to kind of go back and get chemo probably helped a little bit on the Dato trial because you really needed that ADC at that point. But a little unclear why we saw that. So I think somebody that has a high burden of disease, we really need a response. That may be a swaying factor to Dato.
Yeah, I'm really glad that you brought that up because I was certainly struck by the response rates in TROP2-Breast02. And I think for patients with metastatic disease, when we do deal with symptomatic tumor burden, it tends to be fairly rough in triple-negative breast cancer, right? These are when we do see things like ascites and pleural effusions and so forth. Often that's in the setting of triple-negative breast cancer. So I like your point about we have options, and the option to use an agent that has that kind of a response rate in high-burden disease or highly symptomatic disease, I suspect will translate to a quality of life outcome as well, in addition to the efficacy outcome. So, it's interesting you point out that we have options, and I'm a huge proponent, as I know you are too, of mutual decision-making with our patients and really patient-centered care. And so now to be able to walk into that conversation with the patient and say, rather than we have limited treatments and this is what you're going to do, to be able to walk into that conversation and offer more than one choice, but also to be able to point out differences between the two agents, as you've pointed out, the schedule, the adverse effect profile, and the trial design differences, to also give us some guidance in terms of treatment selection, I think is helpful in this setting. So we've seen data now for ADCs, 2 ADCs in the frontline setting for TNBC. Is there still a scenario in which you might consider using chemotherapy?
Oh, that's a great question. Just to set the caveat, we don't have approval yet, so we can't do this tomorrow when we get back home. But, with having these agents, no. For triple-negative breast cancer, I really can't see a reason that I wouldn't use one of these 2 agents in the first-line setting. I think triple negative is a poor prognosis. I'm certainly not saving it for later. I think patients need that PFS benefit in the first line. We know that there's attrition between first line and second line and subsequent lines. Unfortunately, some patients aren't going to make it to second line. And so you really want to give your best drugs first. And the other question or reason would be tolerability. And honestly, we've talked about what we need to watch out for with these two drugs and how to manage it. But really, it is manageable. And the trial data shows us that it's just as easy to manage as chemotherapy, actually probably easier. I think most patients would find an antibody drug conjugate a little bit easier to tolerate than chemo. So I really don't think from an efficacy or a safety issue, there's a reason to not give it.
Yeah, I couldn't agree more. And now that we have TB02, TROP2-Breast02 data for even the rapid progressors, that might be a population where we previously would consider chemotherapy, or we could only consider chemotherapy previously, particularly doublet chemotherapy. But I think that the ability to use an agent with a different mechanism of action, namely an ADC, in that population will be a very attractive treatment approach. But that being said, let me ask you, for your patients that relapse within a year of their adjuvant or neoadjuvant therapy, what now, based on these data, let's say we have approval for these ADCs, what would be your approach for your rapid relapsers?
Yeah, I think I'd still be probably checking the PD-L1. Yep. It really depends on whether they received a Keynote type regimen and already received pembrolizumab. We don't have data that if you relapsed, while on Pembro or within a month of Pembro that you're going to get benefit in the first line. Mm-hmm. But of course there are, some lower risk patients with triple negative breast cancer that wouldn't have qualified to get Pembro in the neoadjuvant setting that do ultimately relapse. And so it would be important to check in that patient. But otherwise, I think for those quick relapsers, the data is very compelling.
Absolutely. So we've concluded ESMO 2025, and I want to get your thoughts on some of the take-home points, the key practice-changing data, just broadly and briefly. I know that's hard to do because we saw a lot of breast cancer data, but what are you taking away from this year's ESMO?
Yeah, I think we touched on it before, but ESMO 2025 was a fantastic ESMO for breast. I came in with high expectations, but I think I left with those expectations really being exceeded. And really uniquely, it was the first meeting in a while that we'd had blockbuster data in all three subtypes of breast cancer, right? So DB05, DB11, for HER2, we had the two TROP2 ASCENT-03 as well as TROP2 in the triple negative, and then in the ER positive, we actually saw data as well. Yes. We saw giredestrant data with everolimus, and then we also saw another G word, gedatolisib, so an IV PI3 kinase inhibitor. So we kind of had two big studies in all of the diseases that really I think most of us are walking away pretty inspired, ready to get home and, get that next step up on the staircase to better outcomes for patients.
Yeah, absolutely. Really, really well said. To see these advances across all subtypes of breast cancer is really encouraging, both in terms of the efficacy, but we've seen a lot of improvements in adverse effect profiles, newer agents that are more tolerable, quality of life improvements. So very exciting. Well, I want to thank you for the discussion today and walking us through some of the triple negative findings and also your larger take-home points. I think it's going to be a very exciting time as we incorporate all of this data.
Yeah, absolutely.